Okay. First of all, I would like to thank the organizers for giving me the opportunity to present to you a really exciting project where we describe an actually novel photochemical mechanism. And so in our group, we are very much interested in the dynamics of various photoreceptors, and we are using various techniques for doing so. And I guess one of our favorite proteins are phytochromes, and I also would like to emphasize why they're so cool. And so you can find phytochromes in fungi, bacteria, and plants. They have biliverdine chromophore bound to them, which gives these organisms this simple two-color vision, which is red light and a far red light. And they can interconvert, they can photoconvert between these two states, which basically tells plants if they are in a shade or if they are in a light. And we heard already yesterday that this is really important also for photosynthesis to know like when you should do that and when the plant should not do it. Once you shine light on them, they undergo really funky conformational changes starting in picosecond range, where there is some changes happening in the chromophore binding pocket. In the nanosecond, microsecond range, you have then here in the so-called tongue region a change from a beta sheet to an alpha helix, which then also then goes up to the, usually you have an output module on top here, to the to a complete opening on millisecond timescales. So, and my talk will focus today on the ultra-fast timescales. So we will mostly see changes in the chromophore binding pocket. Before I show you now some results of our experiments and our data, I just want to highlight these two publications that have been concurrently published in 2022. But they basically show the same results, which is that the rotation upon excitation is a counterclockwise space-conserving mechanism. And if you look really closely at the simulations, you can see that this happens through this like two dihedrals. So yet now the bind goes up and the D-ring can rotate. And back in 2021, which was actually 2 years before my PhD, our group collected very high-quality different electron density maps at SACLA. And very nice, you can see, is that the signal evolves over time. And also, most importantly, it only evolves around the binding pocket. And now I actually want to show you some of our extrapolated structures. So here I just want to highlight this really important hydrogen bonding network that also Carsten was talking about on Tuesday. And now I show you now here our extrapolated structures. So here you can start slowly seeing the D-ring rotation. And so basically you see a little bit a crooking between the B and C ring, and now you have this space-conserving mechanism. And very important is that we have a really nice initial agreement with the QMMM simulations from previous reported studies. And yeah, I also forgot to mention, so it's important the colors of our density maps that you see in this study. Blue means it's negative and gold means it's positive. So it's a little bit different to what Przemysław showed on Tuesday. So please don't get confused by that. So this was really nice to see. But interestingly, what we saw that was not really observed in the QMMM simulations, that our water network, our hydrogen bonding, kind of gets absolutely like rearranged. And here, after already 233 femtoseconds, we see that the hydrogen bonding between this important conserved histidine and we like to call it conserved pyrowater breaks. And if we compare this now to the QMMM simulation, so these are our distances from the experiment, and these are the distances between the histidine and the waters and all the nitrogens around here, there's not really a lot of movement. And this is now the question, what we ask ourselves, like what can cause this rearrangement? And this is kind of where I got a little bit of inspiration from Maria Andrea Mogrinski and Peter Hildebrand back at the Technical University of Berlin. Where they did basically 10 years ago a lot of structural and vibrational characterizations of diverse phytochromes they looked into the binding pockets, they protonated the histidines differently, and then they calculated Raman spectra and compared them to the experiments. And what they basically saw then characterized what the protonation state of this histidine is, or the histidines. So what I was thinking, like maybe this histidine protonation changes upon light activation, which can definitely happen. So, but I don't know actually QM-MM simulations, but I told Sebastian, I don't think we need QM-MM simulations just to look for protonation change. We can use simple molecular dynamic simulations. So here I just want to show you a little bit the overview of what we've been doing. So you start with your refined experimental structures, dark and light. In this case, I used the 3 picosecond structure. You then protonate the histidine in different ways. So you do the delta, epsilon, and double protonated. And also here, I just want to mention the dark state that has been like characterized by the various characterizations from this group here. It was mostly the epsilon dark state, the epsilon protonation in the dark state. So you then run heavy atomers and MD simulations because we are not only interested in all the water movements. You then can calculate water densities from each snapshot of your simulation, and you can ensemble average that. And then you can calculate a different electron density map. So this is a lot of information, so let's look at it a little bit visually. So this is now our heavy atomer strained MD simulation. If you look really closely, you can see that, for example, this here up here is the dark state. And if you look at the simulation down here, the steering is a little bit in a different, it's already a little bit rotated. So this is our excited state. And also here you can see, for example, this change in protonation. We then now can ensemble average the water densities, and we can subtract them from each other and get really beautiful, nice-looking computed different electron density maps. And now our main finding was, and I will take now my time to explain this a little bit, so we can now compare our calculated maps to our experimental maps. So here on the left side, you have now the experiment, and we can compare them both visually, but also quantitatively. So we can calculate the Pearson correlation coefficient between the maps. So what you see up here now is the one with the star means this is our excited state, how it was protonated, so delta, double, or epsilon. And as I said already before, epsilon was the proposed dark state protonation of this histidine. And what we see is that once the histidine actually has a delta protonated, you see a push of the water and also a really good correlation between the experiment and the simulations. So this was really exciting. And independently from that, also our collaborator Janne Ihalainen at the University of Jyväskylä, he did independently of that femtosecond time resolved IR spectroscopy. And this is also now important to understand because Carsten showed on Tuesday how they labeled the chromophore. In this case, we labeled the backbone of our protein. So once you have a different absorbance spectra of the unlabeled protein, and one you have with the labeled protein, and then we can basically take the difference of the different absorbance to give us delta-delta absorbances. So what you should now see is that the biliverdin, the chromophore peaks should cancel out, and you should be only left with protein signal. And what we see now nicely is that we have a shift around the amide bonds, which is really nice. The biliverdin peak kind of like cancels out. It is quite noisy, but something that I just want to point out, we already see a signal after 100 femtoseconds from the protein, which is not from the chromophore. And also, if you look into literature, you would expect actually in this range here, histidine-specific tautomers. So that was really exciting. And before I now come to the conclusion, I also just want to briefly talk about a possible charge transfer. And so when you now excite the biliverdin into the S1 state with time-dependent DFT, you can see that you have a small shift from the C D ring towards the B C ring here. This is also what already Carsten showed, which makes it easier for the D ring to rotate. But if you compared to typical charge transfers on chromophores, this is a very small charge transfer character. And also what the simulation showed is that this is not enough to push the pyrowater out of the vicinity. You need something more extreme. And also, if you look at a little bit into literature and also at mutational studies, is that what it shows is that the histidine 260, it buffers the biliverdin in the ground state. And also most histidine 260 mutants lose the complete photocycle, except actually glutamine, which we believe is maybe has some bulky behavior that it mimics from histidine. So with that, I would like to now come to the conclusion that upon light activation of our chromophore, we see a signal from the chromophore towards our neighboring residues, especially this histidine 260. So this is this electrostatic change that Carsten was talking about, which then changes the chemical environment of our histidine, which then changes the protonation state, and this happens really fast. This happens within 300 femtoseconds, and this is really different from well-studied proton-coupled electron transfer and excited state proton transfers, because these things usually happen on the chromophore, while our, we like to call it remote proton transfer, it happens remotely on a residue. And we also believe this could be maybe a widely used mechanism in other photoreceptor proteins. I would just like to mention photosystem 1, because they also showed that there is a water close to histidine which starts moving upon photoactivation. And with that, I would like to thank my group. I would like to thank my supervisor. I would like to thank everyone who was involved in this project, especially Madan, who is sitting over there. Wei Xiao, who is in Gothenburg at the NMR Center, and also Janne, who performed the IR experiment. And yeah, and I also thank you for your attention.
Thanks a lot for this cool talk. Sure, there's questions.
Oh well.
I saw this one first. So.
I only have a comment.
Yes.
This is extremely exciting, but just because you mentioned photosystem 1,
yeah.
I just want to comment that we've been studying structural dynamics of photosystem 2 for some time now, where some very puzzling fluorescence yield changes are happening after photoexcitation. And we start to believe that it has to do with movement of water on the luminal side of photosystem 2. Okay. But yeah, that's all I've got to say. But yeah, we should follow these channels.
It sounds very exciting.
Yeah. Thank you.
And even though it's just such a small change in water, and you can see that, and that's really exciting, I believe. And you can learn so much just from looking at water movements.
I was just wondering, maybe you said it, but can you go back to the conclusion? So I didn't get, so this new proton on the histidine, does it move from the epsilon side to the delta side, or is this does it come from somewhere else?
So we don't know where this proton is coming from, since our spectroscopy data does not really give any insights on that. We don't really see hydroxide, so if you think if it comes maybe from water somewhere, we don't really see hydroxide bands. So what I actually believe is that it's a tautomerization effect, so that it moves from epsilon to the delta position.